In part 2 of this introduction, we'll talk about the bacteria that's at the center of this paper, Mycobacterium abscessus. So M. abscessus is a gram-positive non-motile rod-shaped bacteria. And many strains of this Mycobacterium, and so Mycobacterium is in the same genus as other pathogenic bacteria such as Mycobacterium tuberculosis and Mycobacterium leprae. Mycobacterium abscessus and the other Mycobacterium are known for being extensively antibiotic resistant. So strains of this bacteria are resistant to many antibiotics. M. abscessus is common in soil and water contaminants, and it causes a number of human infections, including chronic lung infections and soft tissue infections that are shown in these images here from the CDC. So antibiotics are chemicals, drugs that we use to treat infections caused by bacteria. So antibiotics stop these infections by preventing bacteria from growing or causing those bacteria to die. And the way that antibiotics work, the way that they kill or prevent bacteria from growing, is by targeting a number of biochemical pathways inside of bacteria cells. We want those pathways that antibiotics target to be sufficiently different in bacteria as they are in eukaryotic cells so that we don't have side effects. And so 5 of the most common targets for antibiotics are the bacteria cell wall, that layer of peptidoglycan, which human cells do not have, the bacteria cell membrane, which is sufficiently different in bacteria from human cells. Also, DNA and RNA synthesis, protein synthesis, and many important bacteria-specific metabolic pathways. So bacteria resist antibiotics or avoid the effects of the antibiotic drugs through a number of different mechanisms. One of the most common ways is by efflux pumps, which are transmembrane bacterial proteins that pump antibiotics outside the cell. So if they're not in the cell anymore, they can't harm the bacteria. Bacteria also avoid the effects of antibiotics by developing immunity, where the specific protein or enzyme that's targeted by the antibiotics is either changed or no longer used in the bacteria's physiology. Those enzymes that are the target of the antibiotics can also develop mutations so that the antibiotic drug no longer targets the enzyme, but the enzyme is still able to do its important job in the cell. Also, many bacteria produce inactivating enzymes, and those are proteins that are able to degrade or inactivate the antibiotic drug itself. So bacteria evolve antibiotic resistance in the following way. So in a population of millions or billions of bacteria, even though those bacteria replicated their DNA from a single bacteria common ancestor, there will be genetic variants in that bacteria population. Some of those genetic variants might be more resistant to a particular drug than others. They might have small variations in their genetic makeup that make them resistant. Normally, these resistant mutations don't provide any benefit for the bacteria. However, if those bacteria are introduced into a situation where there are antibiotics present, the normal or wild type bacteria will be killed or prevented from growing by the antibiotic. That allows those resistant variants a chance to take over their environment. So this is the concept of survival of the fittest. When there is an environmental condition that promotes the survival of a particular genetic variant, then that genetic variant becomes dominant in the population. So cystic fibrosis in particular is a hotbed for antibiotic-resistant bacteria, and that's because CF patients experience frequent respiratory infections throughout the course of their life. Because they are unable to clear mucus, they develop repeat infections, In order to treat those infections, they are given antibiotics that are necessary to fight back those bacteria. But because they have so many repeat infections, that gives lots of opportunities for resistant bacteria to take up residence within their lungs. Another place where antibiotic resistance is very common is inside of hospitals, where there are many sick patients congregated, often taking antibiotics, either precautionary because they are about to go into surgery or they have weakened immune systems, or because they are sick with bacterial infections. This also gives bacteria that are found within hospitals many opportunities to develop, or it gives antibiotic resistant bacteria within those hospitals many opportunities to become predominant. And so hospitals have to be very careful in managing antibiotic resistant bacteria. So nosocomial diseases is a word that we use for diseases that are acquired in a healthcare setting. And antibiotic resistant bacterial infections are a really big problem in hospitals around the world. One other technique that you should know about when reading this paper is positron emission tomography, or PET-CT scans. So a CT scan is it's a device that uses a rotating X-ray beam to make a cross-section of the inside of someone's body. Right? A PET scan is a modification of the CT scan, where a small amount of radioactive material is put inside of the patient, and that radioactive material is designed to be taken up by certain physiological structures in the body. So it's going to be taken up by certain cells and incorporated into those cells through metabolic reactions. Right? So FDG, which stands for fluorodeoxyglucose, is a molecule that's similar to glucose, which is often used by bacteria and other cells in their metabolism. Right? And it's often used as a radio tracer because it's taken up into cells, cells that are more metabolically active, things like bacteria that might grow much faster, absorb that glucose at a high rate, and so they are more visible in the PET-CT scan. We also use PET-CT scans to identify other metabolically active cells, such as cancer cells. Here are a few questions to consider when you're reading this paper. Was the patient suffering from a bacterial infection in addition to her cystic fibrosis? How many different antibiotics was the patient taking? There's a good figure in this paper that shows all the different compounds that Isabel was taking during the time of the study. What is the mode of action for these antibiotics? Take some time to look these up because the mechanism of action of antibiotics is a really important thing in biochemistry. So was each mode of action similar? Or was Isabel given antibiotics that were targeting multiple different cell processes all at the same time? And why do you think this might have been?